Hello everyone, in this class we'll learn about regulation of blood pressure. The learning objectives of this class would be to describe the mechanisms of regulation of blood pressure, draw a schematic diagram of baroceptor reflex, understand the importance of baroceptor reflex in regulation of blood pressure, and understand the importance of other reflexes in regulation of blood pressure. The blood pressure is regulated normally by short-term mechanisms, intermediate mechanisms, and long-term mechanisms. Here we'll be learning about the short-term regulation and intermediate regulation of blood pressure. Coming to the short-term regulation of blood pressure within seconds to minutes or in an hour is called as short-term regulation of blood pressure. Okay, so there can be neural mechanism, there can be vascular mechanism, there can be hormonal mechanisms. The neural mechanisms mainly consist of autonomic regulation, medullary regulation, hypothalamic, cortical, and reflex regulation. And autonomic could be sympathetic or parasympathetic control, and the reflex regulation are mainly the baroceptor reflex, chemoreceptor reflex, Cushing's reflex, somatosympathetic reflex, atrial stretch reflex, ventricular and pulmonary stretch reflexes, non-physiological chemoreflexes, and Bainbridge reflex. Whereas in vascular mechanism, there is a capillary fluid shift mechanism, stress relaxation, whereas hormonal mechanism consists of catecholamines, renin-angiotensin system, ADH, atrial natriuretic peptide, kallikrein system, histamine, endothelins, EDRF, and adrenomedulin. Where vascular mechanisms, they are important for intermediate regulatory mechanism, whereas the long hormonal mechanism, they are important for long-term regulation of the blood pressure. Neural mechanisms. Coming to the autonomic regulation, the first sympathetic control, the main control mechanism for blood pressure regulation is sympathetic fibers and are the final pathway for all the neural inputs to the blood vessels. There can be a sympathetic vasoconstrictor system and sympathetic vasodilator system. Sympathetic vasoconstrictor system, noradrenergic fibers supplying the blood vessels are vasoconstrictor in nature. Therefore, stimulation of these fibers increases blood pressure. Especially the sympathetic innervation of resistance vessels like arterioles, small vessels, and meta-arterioles are important for the regulation of the blood pressure. Sympathetic vasoconstrictor fibers have a basal rate of tonic discharge, which is essential for maintaining the normal vascular tone. Thus, the vascular sympathetic tone is also known as sympathetic vasoconstrictor tone. Sympathetic stimulation also produces venoconstriction. Venoconstriction increases venous return and cardiac output and shifts blood from the venous compartment to the arterial compartment that aids in increasing blood pressure. Sympathetic stimulation also increases blood pressure by its cardiostimulatory effects and its effect on catecholamine secretion from adrenal medulla. There is also sympathetic vasodilator system, stimulation of sympathetic fibers supplying arteries and arterioles of skeletal muscles produces vasodilatation as they are innervated by sympathetic cholinergic fibers. These fibers originate from the frontal cortex and relay in the hypothalamus and midbrain, and these fibers pass through the medulla without relay in medullary cardiovascular centers and terminate in intermediate lateral gray column of spinal cord. It is to be noted that there is no basal tonic discharge of the sympathetic vasodilator fibers. Then we have a parasympathetic control. The systemic blood vessels are not supplied by parasympathetic fibers. However, vagal stimulation decreases the blood pressure by decreasing heart rate and cardiac output. These are the diagrams showing sympathetic vasoconstriction fibers which are appearing from the intermediolateral horn of the spinal cord. Then they are going to innervate the blood vessels, heart, and adrenal medulla, from where they are going to increase the release of catecholamines. And this diagram showing the sympathetic cholinergic system, where they're going to arise from the frontal cortex and then come to the hypothalamus medulla. So without relying in the medullary regulating center, they directly come to the spinal cord, where they're going to go to the sympathetic ganglion. Then these cholinergic fibers, they're going to innervate the blood vessels, which basically leads to vasodilatation. Then coming to medullary control, cardiovascular centers are mainly located in the medulla. Medullary cardiovascular centers are broadly divided into two: vasomotor center and cardioinhibitory centers. Vasomotor center. This center is present in the rostral ventrolateral medulla, that is RVLM. So this area has a caudal intermediate and rostral part, and accordingly they are called as caudal intermediate and rostral ventrolateral medulla. Though RVLM is the major area, in these group of neurons, the neurons groups are collectively known as vasomotor centers, which directly project to the intermediolateral gray column of the spinal cord via bulbospinal pathway, from where the sympathetic vasoconstriction fibers originate. The neurons of VMC always active and provide tonic excitatory drive to the spinal cord neurons. The basal tonic sympathetic vasoconstriction tone is due to the drive of preganglionic sympathetic neurons by tonic discharge of VMC neurons. This medullary spinal cord cardiovascular axis is the most important pathway for control of blood pressure. As vasomotor center regulates the activity of this pathway, the control of blood pressure by it is called as vasomotor control. Stimulation of vasomotor center causes intensive vasoconstriction and cardioacceleration that increases blood pressure. Inhibition of vasomotor center causes vasodilatation and cardioinhibition that decreases the blood pressure. Influence of VMC on the control of blood pressure is so crucial that loss of this influence, as occurs in following transaction of the spinal cord, results in gross hypotension and acute phase of spinal shock. The vasomotor center receives inputs from the following structures. It receives information from aortic and carotid baroceptors, which are mainly inhibitory to vasomotor center. Then cardiopulmonary baroceptors, again they are inhibitory. Carotid and aortic chemoreceptors, they are excitatory. Ascending pain pathways, again they are excitatory, and ascending pathways carrying proprioceptive information are excitatory. And the lungs via vagus nerve, they are inhibitory, and limbic system and via hypothalamus, usually they are excitatory, but maybe sometime inhibitory. The following are the effects of activation of these inputs. The fibers from the limbic cortex to vasomotor center mediate change in the blood pressure during emotion and excitement. The lung inflation inhibits vasomotor center that produces vasodilatation and decreases blood pressure. Pain and proprioceptive stimuli increase blood pressure by stimulating the vasomotor center. Carotid and aortic chemoreceptors stimulate, whereas baroceptors inhibit the vasomotor center. Hypoxia, carbon dioxide directly stimulate the vasomotor center. Okay, these are some of the inputs to the vasomotor center as we discussed, and ultimately through the intermediolateral horn, it is going to affect the vessels, the heart, and the adrenal medulla to exert its sympathetic effects. Cardioinhibitory centers. Cardioinhibitory center in the medulla is formed by nucleus tractus solitarius, the nucleus ambiguus, and dorsal motor nucleus of vagus. The stimulation of these areas results in bradycardia and decreased cardiac output by two mechanisms. Vagus nerve originates from cardioinhibitory areas, mainly from the nucleus tractus solitarius, which on stimulation activates vagal output that inhibits the heart functions. The NTS inhibits vasomotor center via locally inhibitory interneurons that in turn decreases the sympathetic output. Thus, stimulation of NTS increases the vagal activity and decreases the sympathetic activity via that in turn results in bradycardia, decreased cardiac output, vasodilatation, and decreased blood pressure. So here you can see the NTS, which is also going to act as the sensory or input to the cardiovascular center. Okay, so then activation of NTS, it is going to inhibit the vasomotor center, and it is going to stimulate the nucleus ambiguous, that is cardiac inhibitory center, and it is going to increase the parasympathetic drive to the heart, and it is going to decrease the heart rate and the cardiac output. And also NTS is going to receive the vagal afferents, the local, the feedback from the heart. Hypothalamic control. Stimulation of anterior hypothalamus produces hypotension and bradycardia, and stimulation of posterolateral part of the hypothalamus produces hypotension and tachycardia. Hypothalamus controls cutaneous blood flow that affects the blood pressure. Hypothalamus also mediates the effects of cardiovascular reflexes such as atrial stretch reflex. A reciprocal connection exists between hypothalamus and vasomotor center. Limbic cortex influences vasomotor center via hypothalamus. Hypothalamus also secretes ADH that conserves body water in hypovolemia and hypotension, a mechanism involved in long-term regulation of blood pressure. Cortical control. Cortex does not directly influence the blood pressure. However, stimulation of the motor and premotor cortices usually increase the blood pressure. Sympathetic vasodilator system originates from the frontal cortex. Then coming to the reflex regulation of the blood pressure. Baroceptor reflex. The baroceptor reflex is as it regulates the blood pressure within the seconds. Baroceptor reflex is therefore a life-saving reflex. This is also called baroreflex or synoiotic reflex or Mary's reflex. The receptors for baroceptor reflex are baroreceptors. They detect change in the pressure in the blood vessels and chambers of the heart. Usually baroreceptors are classified into two categories: high pressure and low pressure receptors. High pressure baroreceptors are located in the ventricles and arterial side of the circulation, and low pressure baroreceptors are mainly present in the atria and pulmonary circulation, so which are called as cardiopulmonary baroreceptors. Receptor for baroreceptor reflex are high pressure baroreceptor and that are present in the wall of the carotid, sinus and aortic arch. Carotid sinus is the initial dilated portion of the internal carotid artery at its origin from common carotid artery. These receptors are branched, knobby and intertwined terminals of myelinated nerve fibers. Baroreceptors detect change in pressure in blood vessels in the wall of which they are located. The increase in the blood pressure causes distension of these carotid sinus and aortic arch and stimulates the receptors as they respond to stretch of that organ. So these are the location of the baroreceptors. They are present at the bifurcation of the internal carotid artery in a dilated part. So you can see this is the carotid artery and this is the carotid sinus. The afferent pathways from the carotid sinuses. Ninth cranial nerve, that is glossopharyngeal nerve, is the afferent from the carotid sinus, and the 10th cranial nerve is the afferent from the aortic arch. Fibers from carotid sinus in the glossopharyngeal nerve form a distinct branch called carotid sinus nerve. This is also called as buffer nerve, as it buffers the blood pressure when the blood pressure changes. Fibers in the vagus nerve that carry sensation from the aortic arch form the aortic nerve. However, aortic nerve forms a distinct branch of vagus nerve only in rabbit. Distension of carotid sinus and aortic arch causes stretching of baroreceptors and increases firing in 9th and 10th cranial nerves respectively. Centers for the baroreceptors. Center for baroreceptor reflex are medullary cardiovascular centers. These include vasomotor center and cardioinhibitory centers. Vasomotor centers, as we discussed, vasomotor center is the central sympathetic center as through the bulbospinal pathway, the VMC projects to the intermediolateral gray column of the spinal cord from where the sympathetic fibers originate. Cardioinhibitor centers. Nucleus tractus solitarius is the major cardiac inhibitory center, and this is the main parasympathetic center as vagus nerve originates from nucleus tractus solitarius. Normally, nucleus tractus solitarius inhibits vasomotor center via interneurons. Thus, excitation of cardioinhibitory center stimulates the vagus nerve via inhibition of VMC, which inhibits sympathetic fibers. Okay. So, this is how when the baroceptors are stimulated, the information goes to the nucleus tractus solitarius, where it is going to inhibit the vasomotor centers, and at the same time it is going to excite the cardiac inhibitory centers and increase the parasympathetic activity. And here, because of the inhibition of the vasomotor center, so there will be the vasodilatation, and there will be reduced production of catecholamines from adrenal medulla. Efferent pathways and effector organs. Efferent fibers for baroceptors are sympathetic fibers and vagus nerve. Sympathetic fibers originate from intermediolateral gray column of the spinal cord, which is controlled by vasomotor center. Vagus nerve originates from NTS. Vagus nerve innervates heart, and sympathetic fibers innervate heart and blood vessels. Responses. Responses depend on the nature of change, increase or decrease in the blood pressure. Responses also depend on the degree and the rate of change in the blood pressure. When blood pressure increases, the pressures stimulate the baroceptors in carotid sinus and aortic arch that increases the nerve traffic in 9th and 10th cranial nerves. So when blood pressure increases, there will be stretch of carotid sinus that increases the discharge in 9th and 10th cranial nerves, which stimulates the NTS. There will be activation of the vagus nerve. There will be decreased heart rate and decreased cardiac output. At the same time, there can be inhibition of the vasomotor center, so that decreases the sympathetic outflow, decreases the myocardial contractility, and leads to vasodilatation, which ultimately decreases the blood pressure. This leads to the excitation of NTS in the medulla, which in turn inhibits the vasomotor center via interneurons. Inhibition of vasomotor center decreases the sympathetic output, causes vasodilatation, bradycardia, and decrease in cardiac output and fall in blood pressure. Excitation of NTS also directly inhibits heart by stimulating the vagus nerve. And when blood pressure decreases, the fall in the blood pressure causes less distension of the carotid sinus and aortic arch that decreases the receptor activity and inhibits discharge rate in the afferent nerves. This inhibits nucleus tractus solitarius, that in turn causes disinhibition of the vasomotor center. Thus, vasomotor center is stimulated and that increases the sympathetic discharge and causes vasoconstriction, tachycardia, increased cardiac output, and increase in the blood pressure. Inhibition of NTS also stimulates the heart by inhibiting vagus nerve. Sympathetic activation also promotes release of catecholamines from adrenal medulla that stimulates heart and cause vasoconstriction. So this is when the blood pressure is reduced, there is a less stretch of the carotid sinus that decreases the discharge from the sinus nerve as well as the 10th nerve. There is less stimulation of NTS that causes inhibition of the vagus. There will be increase in the heart rate, whereas there can be disinhibition of the vasomotor center that increases the sympathetic flow, which increases the myocardial contractility and vasoconstriction, ultimately increase the blood pressure. Blood pressure range for baroceptors Baroceptor regulation. Baroceptors regulate the blood pressure in the pressure range of 50 to 200 mm Hg. However, a linear relationship is observed for the change in blood pressure and baroceptor discharge between the pressure range of 70 to 140 mm Hg. No response is detected when blood pressure is less than 50 mm Hg, and no further increase in the response occurs when pressure is more than 200 mm of Hg. There can be different type of responses. Baroceptors respond to change in pulse pressure and change in mean arterial pressure. Accordingly, there are two types of responses: dynamic response and static response. Response to change in the pulse pressure is called as phasic or dynamic response, and the response to change in sustained pressure, that is mean pressure, is called as a tonic or static response. This diagram shows the regulation of the blood pressure by the baroceptors, where there is a linear relationship between the mean arterial pressure and the baroceptor firing, from the 60 to 150 mm of Hg. So there is a linear relation between the mean arterial pressure and the discharge of baroceptors. Okay, so as blood pressure increases, the firing rate is also going to increase proportionally. And this diagram shows the effect of the pulse pressure. When pulse pressure increases, there is also increase in the rate of discharge, and when the pulse pressure is less, obviously there will be less discharge from the synoaortic nerves. And this diagram shows the different levels of mean arterial pressure. When the mean arterial pressure goes on increasing, the rate of discharge in the synoaortic nerve increases. Chemoreceptor reflex. The receptors are here, the chemoreceptors that are located in the carotid bodies and aortic bodies. They respond to change in the chemical composition of blood that occurs in conditions like hypoxia, hypercapnia, and acidosis. The afferents are usually the 9th and 10th cranial nerve, and the center are the medullary respiratory centers. They also project to the cardiovascular centers, especially to the both vasomotor and cardioinhibitory centers. The primary effect of the chemoreceptor stimulation are bradycardia and vasoconstriction. Stimulation of the cardioinhibitory center causes bradycardia, and stimulation of the vasomotor center causes vasoconstriction. However, hypoxia that stimulates the chemoreceptors also causes pulmonary hyperventilation and increases the catecholamine secretion from adrenal medulla, that in turn increases the heart rate. Okay, these are called as secondary effects. Therefore, net effect of stimulation of chemoreceptor is there is no change in the heart rate or mild tachycardia and vasoconstriction that increases the blood pressure. The range of regulation of blood pressure by chemoreceptor reflex is between 40 to 70 mm of Hg. Okay, so this diagram shows the chemoreceptor activity, so where they get stimulated from the aortic body and the carotid body, and they may mainly going to affect the respiratory center, the vasomotor center, as well as cardiac inhibitory centers. On the respiratory center, they cause hypoventilation and tachycardia, and the action on the vasomotor center, they causes the vasoconstriction. There will be release of catecholamines, and the secondary effects like the mild tachycardia and vasoconstriction are caused by the activity of the vasomotor centers. Then Cns Ischemic Response, or also called as Cushing's reflex. In conditions of gross hypotension, as seen in acute severe hemorrhage, blood flow is compromised to the vital organs of the body. Decreased cerebral blood flow produces hypoxia and hypercapnia of the vasomotor center. Direct hypoxia and hypercapnia stimulates vasomotor center to its maximum, which causes intense vasoconstriction, so that raised pressure maintains the minimum flow to the important organs. The reflex response is called as CNS ischemic response. This is activated when blood pressure falls below 40 mm of Hg. This is the last physiological reflex to correct the blood pressure, failure to which it leads to irreversible shock, and that is why it is also called as last ditch response. So when gross hypertension occurs, as in cases of increased intracranial pressure, there will be decreased cerebral blood flow. There is a reduced blood flow to the VMC, and it leads to hypoxia and hypercapnia at VMC, which leads to strong stimulation, causes intense vasoconstriction, and the increased blood pressure at the carotid sinus activates the baroceptor activity, leads to reflex bradycardia. When intracranial pressure is high, as occurs in brain tumor, the blood flow to the vasomotor center decreases due to the compression of the cerebral blood vessels. This causes hypoxic stimulation of vasomotor center and causes intense vasoconstriction that in turn increases blood pressure, and this is known as Cushing's reflex. Increase in blood pressure in carotid sinus activates baroceptor reflex and causes reflex bradycardia. Therefore, bradycardia occurs in brain tumors. Somatosympathetic reflex. The increase in blood pressure in response to stimulation of somatic afferent nerve is called as somatosympathetic reflex. It is activated usually following stimulation of ascending pain fibers. Stimulation of proprioceptive pathway also increases blood pressure. The pressure response is mediated by the vasomotor center that receives excitatory input from ascending sensory pathway in the brainstem. So any pain pathway, proprioceptor pathway, while ascending, they give collateral to the vasomotor center that causes increase the sympathetic outflow, which increases vasoconstriction and the blood pressure. Atrial stretch reflex. Two types of stretch reflexes are present in the atria, type A, and there are two types of receptors, that is type A and type B receptors. Type A receptors are stimulated during atrial systole and type B receptors are stimulated during the peak atrial filling. Increased venous return, as occurs in the fluid retention or increased blood volume, increases the atrial filling that stimulates type B receptors. The responses to increased atrial filling are vasodilatation, decreased blood pressure, and tachycardia. Conversely, decreased ECF volume decreases the activity of the atrial stretch receptors that increase ADH release. ADH in turn causes water retention and increases the blood volume and pressure. Decreased atrial stretch also stimulates sympathetic system that increases renin release, which activates the renin-angiotensin-aldosterone system. Increased venous return causes increased atrial filling, stimulates the type B receptors, which leads to sympathetic inhibition. There can be vasodilatation, decrease in the blood pressure, but there will be increase in the heart rate. Cardiopulmonary stretch reflexes. Cardiopulmonary baroceptors are distributed in the atria, ventricles, and pulmonary vascular bed. The ventricular stretch reflex, when distension of the ventricle due to the excess ventricular filling, as occurs in increased blood volume, results in bradycardia, vasodilatation, and hypotension. Thus, rise in blood pressure is checked. Ventricular stretch reflex also plays an important role in maintaining vagus tone that checks the basal heart rate. Pulmonary stretch reflex. Pulmonary baroceptors are located in the pulmonary arteries. They are stimulated when pulmonary arterial pressure is increased, as occurs in pulmonary hypertension. The responses observed are bradycardia and hypotension. Non-physiological chemoreflexes. Coronary chemoreflex. Chemoreceptors present in the coronary arteries and the ventricles are C fiber endings. The afferents from these chemoreceptors are vagal fibers. Injection of chemicals like capsaicin, veratridine, phenylgonide, and serotonin into the left coronary artery produces hypoventilation, bradycardia, and hypotension. This is called as coronary chemoreflex or Basal reflex. Pulmonary chemoreflex. Injection of above mentioned chemical substances into pulmonary arteries produces similar features like hypoventilation, bradycardia, and hypotension. The responses are blocked by vagotomy, and such responses are observed in pulmonary embolism that produces pulmonary microinfarction. Bainbridge reflex, infusion of saline or transfusion of blood produces tachycardia when the initial heart rate is low. This is called as Bainbridge reflex. The receptors are tachycardia producing atrial receptors located in the atrial wall where vena cava opens into the atria. The reflex is abolished following vagotomy as the responses are mediated by vagal fibers. The tachycardia produced by this reflex competes with the bradycardia produced by baroceptor reflex in response to volume expansion. Infusion of saline increases the venous filling that stimulates the TPR reflex receptors and produces the tachycardia, which is mediated by the 10th nerve. Vascular mechanism. The vascular mechanisms operate within seconds to minutes in alteration of the blood pressure. Capillary fluid shift mechanism. When blood pressure decreases rapidly as an acute hemorrhagic shock, the hydrostatic pressure in the capillary decreases. This causes shift of fluid from interstitial tissue space into the intravascular compartment through capillary membrane, and this is called as capillary fluid shift mechanism. In this mechanism, about 300 ml of interstitial fluid is added to the blood. As a result, circulating blood volume increases and blood pressure returns back to the normal. The reverse mechanism operates when sudden rise in the blood pressure increases the capillary pressure and facilitates capillary filtration. This decreases the blood volume and tends to lower the blood pressure. Stress relaxation. When blood pressure increases abruptly, the blood vessels distend in response to high pressure. This imparts acute stretch on the vessel wall. This smoothens the smooth muscles of the blood vessels relax in response to sudden stretch. This decreases the vascular tone and lowers the pressure. The reverse stress relaxation occurs when blood pressure decreases. Acute fall in the blood pressure reduces normal stretch of the vascular smooth muscle. This in turn causes contraction of the smooth muscle and increases the vascular tone, which increases the blood pressure. This is in brief regarding the regulation of the blood pressure by short-term mechanism and intermediate mechanism. Thank you.